Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, once again to another episode of our show. Again, today, bringing you another really fascinating guest today, involved in research, helping to create a better tomorrow for many people out there. Today, we have the honor of being joined by Dr. Paul Peter Tak, who is President, Chief Executive Officer, and member of the Board of Directors of Condel Therapeutics, which is a biotech company that is developing and commercializing novel viral immunotherapies focused on inducing immunogenic cell death in cancer cells at the site of injection, ultimately with a focus of unmasking various tumor neoantigens within the microenvironment, leading to a systemic, durable immune response against tumors, ultimately with the potential to change disease outcomes in a variety of different indications. Dr. Tak received his medical degree cum laude from the Free University of Amsterdam. He was trained as an internist, a rheumatologist, and an immunologist at Leiden University Medical Center, where he also got his Ph.D. And he has been a clinical associate professor of medicine at University of California, San Diego, as well as professor of medicine and founding chair of the Department of Clinical Immunology and Rheumatology at the Academic Medical Center, University Medical Center Amsterdam. And during that time, he was involved in founding one of his first companies, Arthrogen, a gene therapy biotech company. Dr. Tak has published close to 600 papers in the peer-reviewed journals, received numerous awards, including being elected to the Academy of Medical Sciences. Previously, Dr. Tak served as Senior Vice President, Chief Immunology Officer, and Global Development Leader at GlaxoSmithKline, overseeing a cluster of therapy area units, including Dermatology, Immuno-Inflammation, Infectious Disease, and Oncology, seeing the creation of various new medicines in those portfolios. He was also the president and CEO of Tempero Pharmaceuticals, which was integrated into GSK back in 2015. Dr. Tak has also served as a venture partner at Flagship Pioneering, also as president and CEO of Condel Therapeutics, now Senda Biosciences, sits on a variety of board of directors. We're honored to have him with us today, and a lot of really interesting topics to get into. But Dr. Paul Peter Tak, thank you so much for taking the time to come on the show.
It's a pleasure. Thanks for having me.
It's great having you. As I'm reading through your bio, I was mentioning, you've been involved with, so many different therapeutic segments, many of which, we sort of see in 2023 converging, and whether it's, immuno-oncology or sort of the area of inflammaging or the infectious innate immune connection that we saw the last couple of years with COVID. These areas are coming together, and I'd love to just sort of get your sort of background perspective on what you've seen as you've been involved in these spaces, because we'll get into Condel in a little bit, but talk a little bit about that, because I know you also wrote this really interesting paper in Nature Immunology back in 2021 about breaking down silos, specifically in the autoimmune space. Talk a little broader in terms of some of these other areas, sort of integrating oncology and infectious diseases as well.
Yeah, I'm very happy that you've seen my paper in Nature Immunology, because it's really a kind of perspective of how I think about immunology. And as an immunologist, my biased view, but it's also true actually, is that immunology is like a scientific platform.
Right.
That's relevant for almost every disease area. When you think about autoimmunity or other immune-mediated inflammatory diseases, they're driven by an abnormal immuno-inflammatory system. And often you will need to modulate that and actually dampen the immune response because it detects the patient's own antigens, leading to conditions like psoriasis, inflammatory bowel disease, rheumatoid arthritis, etc., etc. Of course, immunology is very important in the field of infectious diseases. I think about the whole COVID-19 pandemic. I think the general population has been able to learn quite a lot about the interaction between the virus, a microorganism on the one hand, and the response of your immune system on the other, which will determine whether you will get sick or not. And if you get sick, how sick you will get, and whether you will even potentially die because of it. The same is true for bacterial infections, etc., etc. So currently I'm focused on oncology. And here again, it's about the balance between the patient's own immune system and cells that become kind of autonomous, the cancer cells. And there's been a real breakthrough in the field of cancer in the last several years based on the development of, for example, immune checkpoint inhibitors that help activate the patient's own immune system against the tumor cells, CAR T cells, which are modified or engineered cells based on the patient's own immune system that may fight cancer. And in line with this, we are also developing a very innovative new immunotherapy that actually helps to educate the patient's own immune system, how to recognize and subsequently eliminate or kill the tumor cells. So I see this as a platform. Same is true for the field of neuroimmunology. Think of multiple sclerosis, ALS, perhaps even conditions like Alzheimer's disease or other forms of dementia. So it's always about the homeostasis in the body and how does the human body and in particular the immune system interact with cells that become autonomous and start to behave in an abnormal way, or microorganisms. And then there's also the whole world of microorganisms that are our friends that live in our body, which is actually the gut microbiome, the skin microbiome. So I look at the human body in a very holistic way, and that's how I moved from one field to another, where I've always had a laser focus on what are the needs of patients. So that's one. Second, how can we really be super rigorous in terms of applying science to make the best decisions? And third, how can you bring the best people together, make sure that they work together and lead them in the right way so that they advance the science to help to better the life of patients. So from my point of view, I've always done the same in a way, but in different fields.
Absolutely. Yeah, that's what I enjoyed again about reading your bio in that context. And then related to that, and then I'll probably get into Condel in a minute, but one other part of your sort of history in the space, which I found very impressive, is your ability to look at pharmacotherapeutic interventions quite broadly, or look at innovative sort of moieties, because obviously, if this was 30 years ago, we have our small molecules, we have our sort of first generation recombinant proteins and so forth. If I had, brought in, for instance, a bioelectronic modulator of the autonomic nervous system for rheumatoid arthritis to you 25 years ago, you probably throw me out of your office. But nowadays, these things are, part of the pharmacotherapeutic armamentarium. Talk a little bit about that part as well, because I think it's very unique about you that you're not afraid to, and we'll get once again to Condel in a minute, you're not afraid to sort of think of unconventional moieties as therapeutics.
Yeah, this has been the story of my life, and that's just how my brain is wired. I'm quite open-minded about new scientific possibilities, and I'm completely naive, so to say, to modalities, which could be small molecules, biopharmaceuticals, think of antibodies, for example, or cell and gene therapy approaches, or immune checkpoint inhibitors are an example of antibody therapies, or even bioelectronics therapy. So actually, my lab in Amsterdam has played a key role in the discovery of the role of the vagus nerve, which is a nerve originating in the brainstem, in controlling inflammation. And it's a story of serendipity. So if you allow me, I will briefly speak about it, although there's nothing to do with Condel. But it is about serendipity and being open and then finding the best solutions for patients. So I had a company, as you mentioned, when I was in Amsterdam called Arthrogen. We were actually developing a form of gene therapy for patients with chronic forms of arthritis, like rheumatoid arthritis, who had one joint or two joints that were, in spite of optimal systemic treatment, still not doing well. So still painful and swollen, leading to disability. So the question is, what do you do then, right? Do you change the systemic therapy? Maybe the disease will get worse in other joints. So there's a real need, I thought, for better localized treatments in a condition like rheumatoid arthritis, which is a systemic therapy disease. So we started to develop viral therapies to deliver therapeutic genes to the site of inflammation, which is the joint. For in the hands of a rheumatologist like myself, it takes just a few minutes to deliver it into the joint. There's really no issue. There's no place in the body actually that cannot be easily reached with a needle. Non-physicians tend to forget this, but I mean, it may sound scary, but it's very, very well tolerated.
Yeah.
And then in this company, we started the discovery program with another biotech company that people may know, which is called Galapagos, which is especially based in Belgium and the Netherlands. And we designed a screen, an shRNA library, as it's called, to basically knock down thousands of genes in some of the key cells that we took from the joints of patients with active rheumatoid arthritis. And then we knocked them down and looked at key mediators of inflammation, like tumor necrosis factor, interleukin-6, degrading enzymes like matrix metalloproteinases, etc. And we found several hits. That means molecules of interest. And there was one of particular interest, which was actually the so-called alpha-7 nicotinic acetylcholine receptor. Definitely not something I was looking for. Thought, oh, wait a minute, I need to go back to medical school. But what is the role of this alpha-7 receptor for short? Well, it plays a key role in signaling by the vagus nerve. And although we were looking for new targets for in vivo gene therapy, to make a very long story short, and we've published all of that, and the many podcasts that people will be able to find, we actually found that this plays a role in controlling inflammation. So the vagus nerve senses inflammation in the body. It sends a signal to the brain, which leads to a subsequent downstream efferent signal, actually to the spleen, which will ultimately translate into an anti-inflammatory effect in the joint, in rheumatoid arthritis, in the gut, in, let's say, Crohn's disease, etc. So then we thought, well, maybe you can actually replicate this by using an electrical stimulation of the vagus nerve in the neck. First, we did this in animal models. Then I went to the neurosurgeons in Amsterdam. Are you able to implant an electrical device in my patients with therapy-resistant disease? They did, and we found actually that it worked. So now this played a key role in the creation of a new company called Galvani Bioelectronics. Yep. Which is based in London, a joint venture between Verily, think of the world of Alphabet and Google, and GSK. I was on the board of Galvani. It's still in development. And then there's another company in the US called Setpoint Medical, founded by Kevin Tracy, who has been an absolute pioneer in the field of this mechanism in acute models of inflammation. And I was also a consultant to Setpoint. So that's how it all came together. So another point I want to make is that ultimately you need to think about the needs for patients. And what is the real unmet need? Like in chronic inflammatory disease, that will be to get rid of inflammation completely. In other words, to induce a state of remission, absence of inflammation. In most patients, you can only achieve that by combination therapy, by combining, for example, small molecules, tablets that patients can take, with antibodies like the TNF inhibitors, or maybe different approaches like a bioelectronic approach. You implant the vagus nerve stimulator. This has not been approved yet, but is in development. But if approved, that could be a possibility, etc., etc. The same is true in oncology, where Condel is focused on. We've seen a breakthrough, as I mentioned, by the development of the immune checkpoint inhibitors. Jim Allison has played a key role in the discovery of this pathway. Yep. He received the Nobel Prize laureate. He's actually on the research advisory board of Condel and is very, very supportive. But so this is a breakthrough. But now let's look at the patient population. If you take all patients together with solid tumors, then about 1/3 of the patients will respond, but 2/3 of the patients will not respond. So now the question is, how can you convert non-responders into responders? And one possibility would be to combine it with the viral immunotherapy that Condel is developing. The same is true for CAR T cells.
Right.
So CAR T cells, another breakthrough. Carl June at the University of Pennsylvania is like the father of this field. And it works in many conditions. But for example, in pancreatic cancer, area of huge unmet need, the CAR T cells are not really able to get into the pancreatic tumor because there are cells called the stromal cells that create connective tissue. And this connective tissue keeps the CAR T cells that you may administer to the patient, to the circulation, to the blood, they keep them out of the tumor. So then you need to think, how are we going to break down this stromal barrier? And how do we make sure that the CAR T cells, if they arrive at the site of the tumor, stay happy and stay functional so that they can start to kill the tumor cells? And that's where we could help with the viral immunotherapy combined with the CAR T cell therapy. And I was very happy that we've signed a discovery agreement with UPenn, with Neil Shepard and Carl June, to develop exactly this type of combination therapy. So I'm open-minded about modalities. Ultimately, it's about what can we achieve in patients. And often you will need to use combinations together.
Yeah, and I love that theme personally, and the way you're explaining it is oh so elegant. Even and you know we've covered checkpoint inhibitors and CAR T cells with guests on the show, and usually saying, okay, they do a good job, but there's still a large unmet medical need. You just introduced sort of the principle here in terms of novel. Okay, so here we have virus-related immunotherapy that may have an oncolytic component as well as an immunostimulatory component, as well as the ability to go places that other immunotherapies cannot. So you're bringing together, again, you're converging some very important themes here. Let's talk a little bit about this and I guess come down to some of the core projects you have in the pipeline. And I think a great place to start off would be with your CAN 2409, because once again, thinking of this combination theory that you're talking about, here you have you have the herpes simplex virus combined with this interesting thymidine kinase that ultimately, and then you give ganciclovir, which it interacts with that to make it more potent. Talk a little bit, walk us through this one, because clearly it brings in the combination theme, but again, the unique way you think about what a therapeutic could be in this particular case per pancreatic, lung, and prostate cancers.
Yeah, so it's really about what are we actually doing at Condel Therapeutics.
Yeah.
Well, first I should say, again, it starts with the focus on patients. I'm, in the first place, a physician scientist. And my goal is to improve lives of patients. And by doing that, advancing the science and creating value for all the key players, first patients, of course, but also investors, employees, and everyone else. So we are focused on indications, on diseases with a huge unmet need. At this time, that's early, localized, non-metastatic prostate cancer. And interestingly, almost everybody else is focused on late-stage metastatic disease, which I understand, because you get to quicker answers if you focus on late-stage disease. You can, let's say, there are many metastases, you give your new drug, it works, and then you can find out probably within a year, maybe even earlier, whether there's a beneficial effect or not. If you focus on early localized disease, which may often be a slowly growing tumor, then you will need a long follow-up time. So that's why there's very little competition in this field. The good news is Condel Therapeutics has enrolled these patients already in the past. So we have a fully enrolled phase 3 clinical trial, 711 evaluable patients. So this was not a small project. All of that has been done. And we are going to harvest what has been sown in the past. And this study is going to read out at the end of next year. It could be huge news for patients if this is positive and if it will be approved by the regulator. So area of big unmet need, early localized prostate cancer, and then there are different sub-indications in that field. Non-small cell lung cancer patients, that's where we actually focus on patients at the other end of the spectrum. These patients will have had chemotherapy in most cases. They will all have had in our population immune checkpoint inhibitors that we spoke about. This is now standard of care. But although this has led on the group level to improvement, the reality is still that after 1 year of treatment, in 60% of the patients, there will still be progression of the disease. And these patients run out of options. So that's where we try to create synergy, thinking again about combination therapies between continued immune checkpoint inhibitor treatment, but it was actually an inadequate response. So you give non-specific stimulation to the T cells, but then we add CAN-2409 to the regimen, and we give at this time 2 administrations in total to basically vaccinate the patient against the patient's own tumor. Thereby, we teach the immune system, we educate the immune system how to recognize the patient's tumor. That could lead to synergy. And we have very encouraging data based on the first patients that we presented in December during a virtual R&D day. So people can go to the website of Condel Therapeutics and they will find the R&D day with fantastic presentations, starting actually with Jim Allison, the Nobel Prize laureate, and then other key experts in the field who speak about our programs. So non-small cell lung cancer is the second area of huge unmet need that we focused on. Third, pancreatic cancer. People will know that this is still a huge problem. And we have completed the phase 1b clinical trial where we injected CAN-2409 into the tumor, and where we could show that that leads to an increase in the so-called CD8 positive cytotoxic tumor infiltrating lymphocytes at the site of the tumor. And this is a very cold tumor, as it's called, very immunosuppressive. So there are mechanisms that suppress the immune system to do the job of eliminating the tumor cells. So it appears that we can change that, and this has led to the start of a phase 2 clinical trial where we'll have the first data at the end of this year. And then we are focused on another area of huge unmet need, which is brain cancer.
Right.
So we focus on a group of tumors called high-grade glioma. These patients have a poor prognosis. Most of these patients will have a tumor called glioblastoma. And actually, we focus on the population that's even more unfavorable in terms of prognosis, because they have failed already standard of care neurosurgery and chemotherapy and radiotherapy. So we call that recurrent high-grade glioma. And that's where we test CAN-3110, which is a true oncolytic virus. So CAN-2409, the first lead compound basically that's being tested in prostate cancer, pancreatic cancer, and lung cancer, is not an oncolytic virus, but it is a viral immunotherapy that serves to vaccinate the patient against the patient's own tumor. And you don't need to inject all the tumors. And you don't need to do this very often, actually, very frequently. In lung cancer, we give 2 injections at this time in the patient's life. In prostate cancer, in the phase 3 trial, we give 3 injections in total. So you don't need to come back to the hospital all the time to get another administration. And maybe we'll add like a booster, right? Again, think of COVID-19. We may see if a booster is even better, or maybe an annual booster to top up the immune response. But it's basically an off-the-shelf therapy that leads to an individualized response against the patient's own tumor and also the uninjected distant metastases. So we're very excited. And today we announced that we got fast track designation from the FDA, from the regulators, for the development of CAN-2409 in non-small cell lung cancer. So that's also very important from a feasibility quality perspective that may help to also accelerate the timelines in the future. We are not necessarily restricted to these tumors that I just described, because, as I said, we vaccinate the patient against the patient's own tumor. So we could expand into other tumors as well at the right time. We can't do everything at the same time. We're a small biotech company, but we actually do have proof of mechanism in other diseases as well, like retinoblastoma, with tumor micrometastases in the vitreous compartment in the eye of children. We've tested CAN-2409 in pediatric high-grade glioma, so brain cancer in children, in pleural malignancies, so tumors of the pleura, etc. So we believe we could go into other tumors, and that's why we call it a pipeline in a product.
Right.
There's a very big opportunity also from a commercial perspective for the development of CAN-2409. And again, we've proof of mechanism in each indication, not only in mouse models, but actually in patients, in humans with the disease, which increases significantly the probability of success that we will have a medicine at the other end of this whole process. So then CAN-31, yeah, go ahead.
You know, I just was mentioning I was impressed by when you were talking about CAN-3110, how about one of your targets there are specifically nestin expressing cells in glioblastoma, which is this interesting. We talked about that also on a previous show when talking about neural stem cells, but the fact that you're able to target against various, as we said in the intro, the different antigens is pretty cool.
Yeah, so these approaches are quite different, and together we call them viral immunotherapy. So we choose the treatment that may work best for a specific indication for a specific patient. So CAN-2409 would be in principle for all comers, but subsequently we will induce or elicit an immune response that is personalized against the patient's own tumor. So CAN-3110 is a true oncolytic virus. It's an HSV. So it's herpes simplex virus. Right. And whereas everybody else has knocked out a certain gene called the ICP34.5 gene, sorry for all the technical terms here, but everybody else has knocked it out. Why? Because the 34.5 gene, for short, is associated with neurotoxicity. Right. So it makes kind of sense to knock it out, to make it safer. Right. The problem is that if you do that, you will also sacrifice some of the efficacy, because this gene plays a key role in replication of the virus and in the aggressive action of the virus against the tumor cells. So there's, for example, an oncolytic virus that has been approved in the Western world some time ago already. That's called TVEC or Immunology for the treatment of melanoma in the first place. And this is commercialized by Amgen. So it's great that it has been approved. It means that it works, but it's not a huge commercial success. And the reason is that you need to inject many tumors, and you need to do it quite often. And at launch, they missed the endpoint of overall survival, which is very important for patients, of course. They want to live longer with a good quality of life. Right. And I think the reason is that they knocked out this 34.5 gene. So unlike everybody else, we have reinserted one copy of this gene, one allele, under the control of a tumor-specific promoter. That means it will only get activated when this specific promoter is expressed, when it's present. Yep. And the promoter is the nestin promoter. Right. And the nestin molecule plays a role in embryogenesis during the development, the early development of a human being. But then it disappears from most tissues. For example, it's absent from the healthy adult brain, but it is strongly upregulated in high-grade glioma cancer cells in the brain. So that opens the possibility to use CAN-3110 to be super aggressive and replicate at the site of the tumor while sparing the healthy tissue of the brain of the same patient. Of course, that's a beautiful concept, but then it needs to be proven that this is really safe in humans, right? Nobody else has injected such an HSV vector into humans, let alone into the brain of humans. So therefore, we went to completely therapy-resistant disease. Patients who have failed everything and who have basically a few more weeks or a few more months to live. On average, less than 6 to 9 months. And these patients got one injection, a single injection, in a way that was increasing dosages. So this is called a sequential dose escalation clinical trial. And we have now data, and we've presented data now in 41 patients, and we did not observe dose-limiting toxicity. So it works. And actually, you can find CAN3110, the HSV antigen, at the site of the injection. We found it actually in a tumor that was not injected of the same patient, in a patient with so-called multifocal disease, so multiple tumors in the brain, but we could not detect it in the brain tissue in between. So how do we explain that? I won't deny that we were also surprised. It's great, actually.
Yeah.
So the idea is that CAN3110 travels around the brain. It does not replicate, but it does not see nestin in the normal brain tissue.
Right.
But then it arrives in an uninjected tumor. It sees nestin, it starts to replicate again, it starts to attack the tumor cells. That's exactly what we see. We've seen now that the median overall survival in this group with a very unfavorable prognosis is 11.7 months. So that's almost a doubling of what you would expect. And now the next step is we've started to evaluate the effects of multiple injections. Now that we know that it seems safe and well tolerated, especially in light of the severity of this disease, we're now going to ask the question, what happens if you give 2 injections or 3 or 4 up to 6? And the first patient has the first patients are being identified actually, and we will learn a lot from this. We also know that we can induce a CD8 positive T cell response similar to what we've observed for CAN2409 treatment. So you asked me, so what is now your vision for the medicine? Well, of course, ultimately we could go and test any indication that is characterized by nestin overexpression. And we found that there's also nestin expression in melanoma, skin cancer, in gastrointestinal tumors, for example, esophageal cancer, in certain forms of breast cancer, including triple negative breast cancer, in thyroid cancer. So that's where you will have a diagnostic that you could use in the context of precision medicine. And you could imagine that you would test this in patients who have high nestin expression, and nestin expression would be your diagnostic biomarker. So we've not tested this in tumors outside the brain, but it's a very strong rationale to do this at the right time. So here again, we can create what's called a pipeline in a product. But high-grade glioma in itself is already, of course, an incredible opportunity for patients to improve their lives if it works. And we have seen a few transformative case reports already. And then, of course, this is also a very big commercial opportunity for the company. So that's the second investigational medicine.
Excellent. Really.
And then after that, we've created a new discovery platform, which is called Enlighten.
Yeah, let's talk about this. I mean, so Carl June and Bruce Levine are about a mile that way. I'm sitting here in Philadelphia right now, and Penn's right over there. I know you recently announced a relationship with Penn. Talk a little bit about Enlighten and how obviously you talk about nestin and sort of how it works across different tumors, but other interesting neoantigens that you can mention, and if you could say something about the collaboration with those guys, they're pretty cool and local.
No, absolutely. In my previous life, especially at GSK, when I was the Chief Immunology Officer and the Global Head of Development, and also, like say, the super portfolio manager almost, I've given a lot of thought to the question, what can you do to increase your probability of success if you try to discover and develop a medicine? And in the industry, sometimes the negative term is used for that. It's called attrition, right? Think of the wars of attrition. And in other words, how can you reduce attrition? And there are many ways of doing that. And I won't go into too much detail, but I will highlight one criterion to consider, which is to focus early on in discovery on human biology for drug discovery. If you are completely focused on mouse biology, you may be completely wrong. I learned it also in academia, actually, because if you go back to the field of biologics, based on mouse models, people have predicted that you should block IL-1, interleukin-1 in rheumatoid arthritis rather than TNF. But it was actually based on human biology experiments where people at the Kennedy Institute in London at the time took synovial biopsies from the joints, very similar to what I did at Arthrogen actually. And then they put them in culture in vitro, and then they found that actually TNF is a key regulator. So again, example of human biology.
Sure.
So I thought, let's create a systematic approach to discovery of new viral immunotherapies at Condel. And I think we are the first in the industry that has done this, actually. And it starts with a very strong focus on human biology. For example, deep immunophenotyping of human tumors, and human in vitro assays, and human ex vivo assays, etc. That leads to an awful lot of data that you need to analyze in the most sophisticated way possible. In other words, using advanced analytics. So think of computational biology, artificial intelligence, what have you. So that's the second pillar. First is human biology, second is advanced analytics. And that leads to a systematic selection of the genes that you want to deliver to the tumor microenvironment to create synergy with another therapy like CAR T cells or NK cell therapies or immune checkpoint inhibitors or whatever problem you try to solve. And then we use as a vector, as a virus to deliver the genes, again, HSV, human, the herpes simplex virus. Why? Because of its large DNA capacity. So you can insert or clone 5 genes into HSV with a few molecular tricks, potentially even more. And then you can inject it, and we can play at Condel with all kinds of characteristics of HSV. We can make it replication defective or replication competent, like Chem3110 is replication competent. But for many purposes, we would use replication defective approaches, which is also quite safe. We can make it more or less oncolytic. More oncolytic is not necessarily better, because there's a kind of in vivo gene therapy element here. You don't want to kill the tumor cells that you just transduced and that become like little factories in the tumor microenvironment to make the protein that could have a beneficial effect. So we can play with transgene expression levels, etc., etc. So that's the principle. We've unveiled the platform at the time that we also disclosed the discovery collaboration with UPenn. So that's where we use HSV to deliver, when we have finalized the project, the genes encoding, for example, degrading enzymes that will break down the connective tissue, or genes that will give a pro-inflammatory signal to the tumor microenvironment and convert a cold tumor into a hot tumor. You could imagine that you will encode genes that will attract leukocytes or immune cells to the site of the tumor, etc., etc. That's the principle, and that's also where we are open for business with pharmaceutical companies, for example, because we could help to solve their problems by design with this very systematic approach. So I'm very happy that I've been able to recruit a fantastic chief scientific officer to lead this project. I said, well, these are the pillars, and can you create this platform? She's called Francesca Barone, and she and her team have done a fabulous job, actually, to deliver on this. So this is a fantastic and unique platform where we could develop new combination therapies and where we are open for business.
Outstanding. Really outstanding. As you know, you were just mentioning the interesting dynamics of the tumor microenvironment. It got me thinking of you know, and obviously, you know, you're a not really that small a company, but as you were mentioning, there's limited amount of, you can't go after everything. But thinking beyond oncology for a moment, because you know you've published extensively on everything that goes wrong with the immune system in other conditions. So, you know, I was reading this interesting paper you wrote on fibroblast and osteoblast in sort of low-level long-term inflammation, things it does to the joints and bone damage and so forth. It got me thinking a lot of this about this area of inflammaging and senescence, which is kind of hot nowadays. You know, they're not cancer cells, but there's a lot of companies out there that want to figure out how to get rid of them because they like to secrete all these matrix metalloproteinase inhibitor, all the stuff you were just mentioning, and destroying and messing up those microenvironments, making them kind of unhealthy. Any sort of pie in the sky, you know, thinking about, you know, what else these viral immunotherapies could go after besides, you know, once you cure cancer, which it sounds like you're on the way of doing, other possibilities to use them.
Yeah, well, I'm really happy to hear that you also read my other work. And for me, it's all related to the same, right? So it is true actually that in chronic inflammatory diseases that something goes wrong, right? And it goes too far to elaborate how I think about this, but it's a long-term process. It starts with an immune-mediated abnormality. It leads to chronic inflammation. And then actually the chronic inflammation may have an impact on epigenetics. So it will start actually to change the expression levels of certain genes, etc. It may even lead to mutations even in the absence of cancer, including P53 mutations. And then cells like fibroblasts become autonomous drivers of the disease. So even if the original immunological abnormality is gone, these patients may still have long life chronic inflammation. So you need to understand all these different pathways in different stages of the disease. The other way around, when I joined GSK, I joined first as the global head of Immuno-Inflammation. So that was everything in autoimmunity, from idea to discovery to late stage development and beyond in terms of life cycle management. Now companies reorganized again. Pharma companies reorganize always all the time, and they reorganize backwards again. But I thought it was a truly interesting model because I oversaw a completely integrated unit. And then there was also a therapy area unit in oncology and infectious diseases, etc. And I had quite a lot of ideas as an immuno-inflammatory specialist, so to say, about what they could do rather than just combining more and more immune checkpoint inhibitors to give more and more non-specific T cell stimulation. I would predict that many of these programs will be relatively unsuccessful. I said, well, maybe you need to take away the breaks in the tumor microenvironment by starting to interfere with these fibroblasts or stromal cells and the regulatory T cells, the Tregs, and immunosuppressive monocytes and macrophages, etc. And actually, that's the world of the immuno-inflammatory specialist, rather than somebody with an oncology background. And they said, well, that's a great idea, but it's not on strategy. So I went to my boss at the time, who was the global head of R&D of GSK, and said, we are missing a trick as a company. And that's why we reorganized and why we created an immunological cluster of therapy area units that I started to lead. And I recruited my successor as the new head of immuno-inflammation reporting to me. And the head of oncology also reported to me. So we could start to integrate this. And I think it led to quite a lot of innovation. So where could this go next? Well, I won't go anywhere because I'm happy at Condel Therapeutics and we're not done yet. So of course our ambition is very high to have a huge impact. But all of that may play out in the next few years actually. In the near term, we are actually until recently unknown biotech company, but with fabulous science at this time and a fantastic research advisory board that supports us and validates us. And real data in patients. I mean, again, that signals the probability of success, right? If you see all these data consistently, then you need to look at the totality of the evidence. But one day, let's imagine that everything goes great and I have a lot of energy to do something else. Then I've always been wrong about my personal predictions, so I need to be careful now. But one field that I would definitely consider is the field of neuroimmunology.
Okay.
I think I've been intimately involved in the revolution in autoimmunity, especially in rheumatology, but also to a certain extent in dermatology and gastroenterology. Then, there's still a big unmet need. But then the next big revolution was actually in oncology. We're in the middle of it. We're not done yet. Others actually shown proof of mechanism with the immune checkpoint inhibitor that if you are starting to modulate the immune system in the right way, you can really cure patients. Now the question is, how can we cure all patients? We're not there yet, still a lot to do. But then the big revolution that's needed will be in the field of neuroscience. Think of Alzheimer's disease, Parkinson's disease, of course, all the other neurodegenerative diseases like ALS, multiple sclerosis, etc., etc. I think we're starting to learn much more. In the past, the brain was really difficult to access, but we learned so much using modern and highly innovative imaging techniques. But you can even take biopsies from the brain. It sounded even scary to me. But actually, in the clinical trial that we are doing with CAN 3110, there are now serial biopsies that we will learn from the tissue response to CAN3110 in the brain, in the tumor. And the neurosurgeons in the best centers of the world assure me that this is possible. Of course, very small biopsies. So we will learn a lot, and I think the immune system will definitely be shown to play a key role in many of these conditions. And hopefully I or definitely others will play a key role one day in helping patients to live a better life, also with these conditions, and with a better quality of life as well.
Excellent, really excellent. So you have, you're a clinician, a researcher, you've been a big pharma executive, you're a biotech entrepreneur. However, in the bio, I also mentioned that in addition to, I neglected to mention, in addition to you being a venture partner, flagship pioneering, you've also been on the scientific advisory board of Index Life Sciences, Fund 6, and Medici Venture Funds. Say a couple words about your venture capital experience, if you would, because that's a whole other animal, let's say. And I just love to hear, how, again, as you've seen things evolving in pharma and biotech, it seems interesting to me, at least, the last time I raised venture capital was 20 some odd years ago. It seems like there's a lot of billion-dollar Series A rounds happening nowadays, which is just kind of strange. But anyway, talk a little bit about what's going on there.
Yeah, so for me, I'm a curious person. So I always want to learn, and I'm very much focused with a laser focus on ultimately improving the lives of patients, and thereby I try to have a little contribution to society. But so there are different roles that you can play, like being the physician, then being the scientist, and develop actually the treatments of tomorrow. Right. Then learn about leadership to really amplify the impact that you can have. So when I moved from academia to big pharma, I thought, well, I was very close to my patients, but I thought, imagine that you make a medicine for, let's say, type 1 diabetes or whatever. You will have an impact on millions of people. So I learned a lot about leadership. Then, of course, you learn that capital is a key tool actually to be successful. And even if you have a very big budget, as I oversaw at GSK, huge budget, then still there's always the basic law of economics of scarcity, right? So you will always need to prioritize, even if you have a multi-billion budget. It's the same whether you lead a small or a big enterprise. And how do you leverage that capital in the best way possible? So for me, it was a truly interesting experience to be very close to Medici at the time. And there was a big fund that was created by Medici together with Janssen and GSK. And that's how I got involved in the SAB, overseeing that big fund. But later, in a very different model, on top of my job as the CEO of Condel Therapeutics, I was a venture partner at Flagship, and I tried to contribute to some of the other companies there, like Omega Therapeutics. But I also learned quite a lot about their model, right? Like Flagship Pioneering, of course, created Moderna, so now they're even more world famous. Yeah. And they are very thoughtful about the creation of discovery platforms. I won't deny that Condel's Enlighten platform is to a certain extent inspired by this experience that I had at Flagship Pioneer. But then I've combined it with my real drug discovery experience at GSK. So now we are facing, of course, a global economic crisis. Based on many reasons, and there are many people who can speak about this in a much more eloquent way than I, but of course it is related to the COVID-19 pandemic, which has led to government spending huge money, right? And I don't have a judgment here, but it has led to ultimately high inflation levels. There were huge supply issues in the factory of the world, which is China, and all of this had a big impact. And of course, there's a big war in Europe. And other factors. And then there's another thing that during the pandemic, the whole biotech world has seen a real hype. And I think ultimately there's not enough money for all the companies that have been created. And although this is going to be very painful, in a way, this may be a healthy correction. Because there are companies that are very much hyped, and ultimately it should all be based on the probability that you will develop real medicines that will be sold in the marketplace and that create real value for investors, rather than beautiful ideas with fantastic slide decks and great narratives. You also need that, but ultimately you need real data in real patients that will lead to real medicine. So I think we may see a kind of healthy correction, and I would hope that the companies that have molecules in the clinic that look really promising based on the results of clinical trials will be the survivors of this carnage that we might see.
Excellent message. Absolutely. Paul Peter, any while we have you, I know we're coming at the top of the hour now, but any other things happening for 2023 in the coming months, conferences you're going to be presenting at, presentations, places that we can see you, meet you, listen to you, anything else on the calendar that you want to mention, please take the floor.
Yeah, so we will announce and we will be clear about all the meetings that we attend and where we will present. We've been pretty active in the last year since I joined, and we will continue to do that. I will just summarize the key data, the key inflection points this year. So first, we expect data in non-small cell lung cancer. I'm super excited about this clinical trial. It's open label in a therapy-resistant population. As I mentioned, these patients will have failed immune checkpoint inhibitors. They have an inadequate response to immune checkpoint inhibitor treatment. About 90% will also have seen chemotherapy. And then in most of these patients, the tumors start to grow. And what we've shown so far, and you can see it on the R&D Day presentation from December on the website, that in most patients, we are able to convert progressive disease, so the tumor is growing, into stable disease. And we are not suggesting that that's going to be the regulatory endpoint of the phase 3 trial in the future, but it is very strong biological proof of mechanism that actually we're changing the tumor growth trajectory. And we would expect that this translates into improved progression-free survival. And indeed, initial data support the view that we have improved progression-free survival. We will have more mature data in Q3 of this year. And then maybe we start to see the first patients, like case report level, where we can start to think about overall survival, because that's the ultimate goal. The patients need to live long enough to have these overall survival data. So for most patients, that will take another year. But we expect new progression-free survival data in Q3, a bit more patients, and more immunological data. For each trial that we do, I apply the principle of what I call experimental medicine, so deep mechanistic clinical trials, where you try to learn as much as possible. And that also validates the clinical findings. Then in pancreatic cancer, we expect new data, initial data from the phase 2A clinical trial in Q4. Next year, Q4, the readout of 2 clinical trials in prostate cancer, early prostate cancer, a phase 2 in the so-called active surveillance population, and the phase 3 clinical trial that could be a registrational clinical trial in patients with intermediate to high-risk prostate cancer, where we combine our therapy with radiotherapy, standard of care, plus or minus hormonal therapy, to basically increase the number of patients, the percentage of patients that will achieve a state of disease-free survival. So no evidence of cancer anymore. That's what you need to aim for. That I called remission in autoimmune disease. Right? That's what we need to try to achieve.
Yep.
Then we expect more data based on the Enlighten platform. We have not announced the timelines for a candidate selected molecule, but we're making good scientific progress. I think these are the key highlights. It's not impossible because we are so focused on operational excellence that we might over-deliver a little bit and that we might have more data as well.
It's not just an impressive portfolio, but as you were just saying, in terms of operational excellence, it's so impressive the way you're executing on it. So I'm rooting you on and just really wishing you the best with all of this. And I hope we'll do a follow-up episode next year when some of these trials wrap up. But really, really great work you're doing. Again, for everybody that's going to be listening to this particular episode of our show across the podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Paul Peter Tak, President, Chief Executive Officer, Board Member of Condel Therapeutics, doing really amazing things to develop and commercialize these novel viral immunotherapies to improve the lives of cancer patients everywhere. Really Great story. I want to thank you, Polpi, for taking the time out of your schedule to come educate us on this novel area of pharmacotherapeutics. Obviously, thank you for everything you're doing for cancer patients. And as we like to say on our show here, thanks for creating a better tomorrow for so many people via what you do. Really such an impressive story.
Well, thank you very much for giving me the opportunity and for a great discussion and great questions. Thank you so much.
Great having you.